Hello, my name is Tyler Johns and I'm a first-year microbiology student here at Cal Poly. The name of my study is the effects of sunlight and foliage density on the growth of trees. This study examined if foliage density and sunlight affects the growth of trees. So how do trees grow? Trees grow by using photosynthesis, which is an important process that allows trees to use their leaves to absorb the sun's energy and convert it into chemical energy for both immediate and later tree growth. Without this conversion as an energy source, a tree cannot grow. How exactly do we measure how much sunlight a tree absorbs through its leaves? We use foliage density, which is the total leaf surface area per unit volume of space. How do we measure a tree's growth? We can measure a tree's growth by using the DBH of a tree, or better known as diameter at breast height. We use the DBH of the tree to estimate the volume, biomass, and carbon storage of the tree. Here are the hypotheses and predictions drawn from this study. The null hypothesis is foliage density and sunlight do not affect the DBH of the tree, and the increase in both of these variables will not lead to an increase in the DBH of the tree. The alternative hypothesis is if foliage density and sunlight affect the DBH of the tree, then the DBH of the tree will increase as the density in sunlight increases, because more sunlight allows for better growth of the tree, and high density displays an abundance of nutrient resource availability. It can be predicted that as sunlight increases, the DBH of the tree will also increase, and as density increases, the DBH of the tree will also increase. To come to the conclusion that sunlight and density have an inverse relationship with the DBH of a tree, and as sunlight and density both increase, the DBH of the tree will also increase. This study was conducted on 35 trees with varied DBH of tree sizes, and were sampled at random. The variety of DBH of tree sizes was an important factor in determining if there was a correlation between sunlight exposure, foliage density, and the growth of the tree. To measure the sunlight exposure on a tree, a picture frame of 10 by 15 inches with a 4 by 4 inch circular hole was held by a person who was faced away from the tree and backed into the tree. They proceeded to lift the picture frame upwards to ensure it was towards the sun. A piece of paper was then held by another person under the picture frame to measure the area of the shape the sunlight was producing on the paper. Calculations were then made to determine the percentage of light coming through. Then every leaf on the tree was counted to determine the foliage density. This was acquired by calculating the total leaf surface area. Finally, the tree was then measured with a measuring tape. With the constant measurement of 4.5 feet from the ground to up the trunk of the tree, the circumference was then obtained at exactly the 4.5 feet mark, then divided by pi, 3.14, to get the DBH of the tree. After, the data was recorded and analyzed with graphs and statistical tests using RStudio. Here are the results from the recorded data. Figure 1 shows a scatter plot comparing the amount of sun exposure to the diameter of breast height. Figure 2 is another scatter plot comparing the foliage density to the diameter of breast height. And figure 3 is another scatter plot comparing the amount of sun exposure with values of 0.00, 0.25, 0.50, and 0.75 to density. Figure 1 was the only singular positive correlation in this study. The correlation and linear model test performed in Figure 1 produced a p-value well under 0.01, which gives us strong evidence against the null hypothesis. However, the adjusted R-squared statistic did not produce a significant linear relationship at the alpha level 0.5. This aligns with the alternative hypothesis. As sunlight increases, the DBH of the tree will also increase, because we see a positive correlation between both variables. However, it also goes against the alternative hypothesis by not having a linear relationship between when one variable increases, the other variable increases as well. This is likely due to the fact that sunlight is a major component in photosynthesis. Trees are directly impacted by the amount of sunlight that hits their leaves. Using that energy to create sugars that allow the tree to survive and grow. However, the primary function of photosynthesis is to convert solar energy into chemical energy, glucose and oxygen, and store that chemical energy for future use to provide nutrients to the tree, with the growth of the tree taking a less important secondary role in its anatomy. The remaining results, figures 2 and 3, produced weak negative correlations with no significant linear relationships between figure 2, foliage density and the DBH of the tree, and figure 3, sunlight and foliage density. Both figures produced p-values of 0.1493 and 0.0932, which is less than or greater than 0.1, and consequently provides weak to no evidence towards rejecting the null hypothesis. With weak to no evidence against the null hypothesis, we cannot reject the null hypothesis and support the alternative hypothesis. This experiment was not consistent with the results from a similar study, Adaptation to Sun and Shade, a whole plant's perspective. The 1988 study finds that photosynthesis is directly impacted by the amount of sunlight that hits a tree's leaves. More specifically, is not consistent with the dependent relationship between sunlight and foliage density presented in the study. This research can further be investigated, making drastic changes to the method of calculating sunlight. Since the experiment's methods of calculating the amount of sunlight the tree as a whole received instead of calculating the sunlight exposure of each individual leaf received, it is inconclusive and fails to accurately report and analyze the relationship between sunlight and foliage density, which is the total surface area of a tree leaf. This could also expose a pattern of inaccurate data for correlation tests and linear models due to the possibility of each leaf not receiving equal amounts of sun exposure. Another method that can be investigated further is the sampling method used. Instead of using a random sampling method, a cluster sampling method can be used to produce a better representation of the observational unit. Thank you.